Welcome to CivilizationX. A petri dish learned Pong. It used 800,000 living human neurons wired into electrodes. In 2022, Cortical Labs showed those cells could change their firing to keep a digital paddle aligned with a moving ball. The anomaly is not that biology computes. It is that detached human neurons outside any brain learned a goal-directed task inside a machine. On Thursday night, a man pauses outside his mother's bedroom and hears pill bottles click again. Her hands hesitate before they obey. That is the real stake. The human brain runs on about 20 watts, yet reaches roughly exaflop-class performance. Frontier needs about 1 million times more power and more than 680 square meters to reach that class. If computing stays that expensive, tools for decoding Alzheimer's, Parkinson's, and damaged neural circuits stay slow, scarce, and out of reach. The system was called Dish Brain. Human and mouse neurons were grown on a microelectrode array that could both stimulate and record them. As Cortical Labs' Brett Kagan put it, "We have shown we can interact with living biological neurons in such a way that compels them to modify their activity, leading to something that resembles intelligence." What matters here is not a stunt with a video game. The hard question is why living neurons can learn from tiny amounts of data. Where silicon systems often need vast training runs, huge power budgets, and rigid optimization. That answer may reshape both computing and brain medicine. Brain organoids are three-dimensional clusters grown from stem cells. They contain neurons and support cells developing together, which matters because real computation in nervous tissue depends on cell-to-cell signaling, not isolated spikes. Jason Chiu said the advantage is that multiple cell types mature together, allowing relevant communication. Electrodes then become the interface. They write pattern signals into the tissue, read electrical responses out, and close the loop through software. In Dish Brain, the loop was framed by the free energy principle: reduce unpredictable input, and the network self-organizes toward more adaptive firing patterns. Here is the counterintuitive part. The neurons were not pre-programmed with rules for Pong. They received structured stimulation representing the game state, and their output shifted the paddle. When their activity produced more stable outcomes, the incoming signals became more predictable. When they failed, the system delivered noisier feedback. Over time, the culture adapted. Moeen Kajihanejad called it the first head-to-head benchmark against deep reinforcement learning under equivalent sampling constraints. In limited data conditions, Dish Brain learned faster and more efficiently than DQN, A2C, and PPO. That is the real rupture. Sample efficiency, not raw scale, is where biology looks stronger. The field did not stop at Pong. In 2023, Indiana University's BrainAware combined organoids with conventional hardware for speech recognition and nonlinear equation prediction, cutting training time by 90% versus silicon-only systems. In July 2024, Meta Bach from Tianjin University and Southern University of Science and Technology let lab-grown brain tissue control robots for obstacle navigation and object gripping after simulation training. By 2025, Final Spark's neural platform was renting remote access to thousands of organoids, each linked to 8 electrodes, for about $1,000 per month. Cortical Labs also released CL1, a commercial biological computer merging human stem cell neurons with silicon, and pushed latency below a millisecond, down from Dish Brain's roughly 5 milliseconds. Then, the interface layer advanced again. BISC, described in December 2025, packed 65,536 electrodes and 1,024 simultaneous recording channels into a wireless implant about 3 cubic millimeters in volume. Andreas Tolia said it turns the cortical surface into a high-bandwidth read-write portal. In April 2026, Northwestern engineers printed artificial neurons that generated lifelike electrical signals and directly activated living brain cells in mouse tissue. The boundary between electronics and neural matter is getting thinner. So what has this achieved? Not a conscious machine, not a replacement for silicon. It has shown that living neural tissue can be a usable computing substrate for low data learning, pattern recognition, prediction, and control. It also offers a testbed for diseases that make a mother's hand pause before it moves. But the engineering gap is still severe. Organoids are fragile, variable, hard to benchmark, and difficult to scale reproducibly across labs. The near-term target is hybrid systems, not biological laptops. Use neurons where adaptation and energy efficiency matter most. And let silicon handle storage, translation, and control. That could accelerate drug screening, neural prosthetics, and low-power machine learning within 5 to 15 years. But solving that creates a different problem: donor consent, mental privacy, cognitive liberty, human-animal chimera ethics, and a new divide between labs that can access these systems and those that cannot. A petri dish learned Pong because electrodes gave living neurons a world they could push back on. Now, that same logic is moving into commercial platforms: robotic control, printed neural interfaces, and direct brain links. The unresolved line is no longer technical alone. As organoid computing systems become larger and more interactive, what measurable biological threshold would let scientists distinguish complex adaptive learning from the earliest signs of sentience before these platforms are deployed at scale? The constraint is real. The data exists. Whether the systems built around it can act on it at scale, that is the next engineering problem.